to be introducing Ella Striem-Amit. She earned her PhD from Hebrew University in 2014. She's therefore a very young scholar by our standards. She then did a postdoc and fellowship at Harvard, and is now at Georgetown University in Washington, D.C. in the Department of Neuroscience. Ella studies how sensory experience changes and shapes the development of the brain. And she does that by looking at highly unusual populations, like the deaf, and the blind, and as we'll hear today, people born without any hands. What happens to the development of the brain when you don't have that normal sensory motor input? She's just set up a lab at Georgetown. She did that since 2019. There are already 10 publications that have come out of that lab, according to Google Scholar, which I checked this morning. And their importance is signaled by the fact that they're in places like Current Biology, Proceedings of the National Academy of Sciences, Nature Communications, and Scientific Reports, which are the places that scientists envy being able to publish. So over to Ella to tell us about what happens to the brain when you don't have any hands.
Thanks, Daphne and Linda, for inviting me and for this very generous introduction. Let's start by sharing my screen. And by figuring out again how to add the video panel so I can see you all. So thank you again for the invitation. I'll try to tell you a bit about what we've learned about the brain plasticity and development from studying people born without hands. And the magic really occurs, or the big question really is to us, how is it that we take something that is essentially a fertilized egg and we get to develop it through only 9 months and then a few years onto a fully intact cognitive sensory motor system that can do all the things that we as fully functioning adults can do, right? What is it that drives brain organization and how does it adjust to its environment? Assuming that there are multiple backgrounds in the audience, I'll start with some basic facts about how the brain is organized. Our brain is comprised of millions of neurons that are organized in a meaningful way such that different parts of the brain have different functions. It's the principle of functional distribution, and we have a big chunk of the brain. I hope you can see my mouse moving.
So, Ella, just before you go on, you have two big black boxes on top of your slides. That one, yeah, and one over to on the right side. Yeah, that we got rid of that one. How about that one? Your mouse is on now.
I would kind of like to keep you all with me, if possible. I don't know how to make it so. Oh, give me 1 second. Let's do.
It just was blocking what you were pointing at us to look at.
Does this help?
Yeah, that's fine. You've got two black.
Do I still have the black boxes for you?
Yes, you do.
Oh, darn.
And as you click the screen, they move around. Now it's over on the left.
I have moved it to the left. I'm trying to find a place where I can still have you with me so I can see your faces while I give the talk. And even though I'm just sharing the slides, it's still showing you all of that, which is interesting.
Okay, so now the black box is towards the bottom. It's not blocking anything you're pointing out. It's kind of interesting. I wonder how our brains are going to reorganize. It's changing size. It's jumping around the screen.
No, this is all my doing. This is not, okay. I'll just have Daphne here in my corner. Daphne, you're with me, okay?
That's perfect.
My marker of human existence in the audience.
Well, anyway, they're the little black squares in the upper left corner and the long rectangular ones at the top. They're not blocking anything you're going to show us on the slide.
No, they will not.
Okay, and yes, we can see your pointer.
Okay, marvelous. Thank you for telling me what isn't working. I appreciate that. So when we're talking about brain organization, we have big chunks of cortex that are dedicated to different functions. We have the visual cortex at the back of the brain, the sensory motor cortex over here up towards the top of our head, the auditory cortex down here in green. So different brain regions perform different roles. And if we're talking about sensory motor cortex, which is what we ought to be talking about when we're talking about hand representations, I can cut open that part of the brain in this way and show you what the motor cortex looks like if we slice our brain in this direction. What you're seeing here, I hope, is the different body parts that are mapped onto those parts of the cortex, of the primary motor cortex. And this is an organization that's called somatotopic. It's adjacent body parts that are mapped onto adjacent parts of our cortex. So we have a representation of the face, a representation of the hand, the arm, the body, all the way to the foot. And what you might already notice in this picture is that areas that are that we have a need or use of high resolution touch and motor control of, such as the hand and the lips and the mouth and the vocal cords, have an increased side of a representation. They are overrepresented in cortex. And this absurd looking little man is called the motor homunculus. It represents the areas, the size of the areas of his body represent the magnification factor on cortex. So you can see that the hands are hugely overrepresented, and so is the mouth. But things like the arm is not as highly represented. And when we talk about hand representations, we actually need to extend beyond the primary sensory motor cortex to adjacent regions that have other representations and that care about other parts of things that we do with our hands. If we are looking here on the top part for perception, we're looking at blue areas of the brain that are areas that are activated or care about when we perceive other people perform hand and arm movements. So you can see, you don't need to know where this is in the brain. All you have to notice is just how much blue there is out there, how much of our cortex is representing the hands. And the same goes to on the lower part here, all the areas highlighted in light green that are areas that are engaged in executing hand actions. So overall, we have really large parts of our brain that represent or are engaged in either perceiving or acting with our hands. And the question is, what do all these regions do when they are not employed? If we take this sensory motor strip of cortex and we take out the hands in people born without hands, what do all these areas do? Do they even function? Do they have another functional role? And the best way to try and assess what they could be doing is to try and see what people born without hands do or can do in order to compensate for their disability. So if my slides will move, I'll show you what these people do and can do. So this is one of our participants that we study. And much like our other participants, she is a very proficient foot user. She uses her feet as if they were hands. And she can do anything with her foot that we can do with our hand. There is really essentially no limitation in her daily life. She can text, she can type, she can draw, she can carve pumpkins. She drives a car. There really is, and there isn't anything that she can't do with her feet. And that tells us, that maybe those hand areas are dedicated not just to the hands, but they allow us to compensate by doing the same types of actions with other body parts, right? It brings about the question whether those hand areas really are dedicated to the hands, or whether they can compensate in a way that makes sense for her to support all those foot actions that we see here. Now, in order to test what other roles they can have, we wanted to see, first of all, what are those hand-related areas involved in people born without hands, and importantly, whether these roles are shared with us, with people who have not lost their hands, who have grown up with hands, or whether this is something unique about their brain that allows them for compensation, whether it's compensatory plasticity, or whether this is something that we all could, in essence, do. So why should we care? Why should we care about how hand areas are involved in people born without hands? The clinical side of it is that a lot of people actually can and do lose either their hands themselves or their hand function over their lifetimes. And this is less of a common issue for amputation. Amputation is not a large, people with amputations is not a high population. It's not a large population, especially if we're talking about upper limbs. There is a larger risk of lower limb amputation associated with diabetes. But stroke really often hinders hand and upper limb function. And because we are talking about a lifetime risk of stroke that is largely increasing given the lifestyles and the long lives that we now have, and so many of the stroke survivors have upper limb disability, it really becomes important to understand how the brain controls hand action and what compensatory strategies or techniques or plasticity can exist, and what other tools we can develop if we understand how the brain represents actions. And that can drive the development of better brain-machine interfaces or hand prosthesis. On the theoretical side of why should we care, personally, I find that congenital limb loss, much like congenital deprivation of other things, is a really fascinating model to try and get at the balance of nature and nurture on brain development. Because on one hand, we really know that the brains are way more similar in identical versus fraternal twins. So we know that there are genetic components in play in how our brains develop and organize. But on the other hand, there are multiple studies, including some pioneering works of Daphne herself, that show that experience plays a crucial role in organizing the brain and fine-tuning it towards intact function. And the complete absence of experience of things like hands from birth can really show us what about brain development is dependent or independent of having that experience of having control of one's hands. Additionally, when we look at models of congenital deprivation or congenital loss, it really brings about evidence for the maximal plasticity possible in the human brain, right? We know that our brains are way more plastic in early development. Again, some of Daphne's works. But we also know that plasticity diminishes over our lifetime, and in adulthood, it is way more limited. So these models of congenital loss kind of illustrate just how plastic the human brain can be. If there's a type of plasticity that we cannot observe, even in people born completely without hands, who have had a lifelong experience of using their feet, we probably won't expect to see that much plasticity following stroke later in life, in aging. So what my lab does, as Daphne introduced, is to study different models of congenital sensory and motor deprivation. And we study, sadly, very small samples of those very clean models of people born deaf, blind, or without hands to uniquely address those questions, those causal questions of the dependence and of sensory motor experience. And today I'm just presenting the handlessness works, but I'm happy to answer questions more broadly about the effects of congenital blindness or congenital deafness on the brain. Now, back to our questions. What happens to the hand sensory motor cortex when one is born without hands? So with these people, we are answering if motor experience is crucial for hand-related brain functions. And to what extent is that organization specific to the hands? Are the same areas lighting up when they perform foot actions as when we perform hand actions? And the people we study are, again, a very small sample of a very clean model of developmental loss. So these are people born without arms or hands on both sides. They are all right-footed, and none of them have any phantom hands or phantom arms, which is something that occurs following amputation, but doesn't seem to occur nearly at all with congenital loss. Now, as I showed you before, I'm going to be showing a lot of brain pictures today. For the most part, you don't really need to care where things are in the brain. The point of me showing you these two pictures of hand reaching and hand grasping is to show that in those association motor cortex, beyond the primary motor strip, there are different areas dedicated to different hand actions, such that there are different areas that are recruited and are functional when we perform a reaching action, as opposed to when we perform a grasping or manipulation of action of different objects. And using people born without hands, we're trying to understand whether these regions are really organized in their core as hand areas that have this sub-specialization for different hand actions, or whether their core principle of organization is for the motor task, reaching or grasping or tool use or manipulation, and just incidentally or partly incidentally, they are used by us mostly with our hands. In other words, do those association motor areas have an abstract representation of the action that doesn't care what body part implements it? Do they have an abstract motor plan that can be implemented when we reach with our hands, with our mouths, with our feet? As any of you who's been carrying groceries, you would know, at some point you have so much things in your hands. If you're opening your door with your mouth or with your foot, or you're pushing the door with your foot, we can perform a lot of our actions without engaging our hands. And the question is, do those actions have the same motor plans with the same neural substrates as hand actions? Now, when we're talking about these general principles of organization, is this area abstracted, doesn't care what body part performs it, we always want to draw on a conjunction of consistency of the findings across healthy controls and individuals born without hands. And the reason is that if we, in controls, find that these areas represents the hands and they represent the foot, in us, this could just stem from imagining our hands, right? We could be imagining actively, oh, how would I do this with a hand? Let me implement this with my foot. This can't be said in people born without hands, but if we only find it in people born without hands using their feet, it could be something unique to them, something unique about their brain plasticity. So looking for things in the conjunction of typically developed individuals activating both different body parts to perform the same actions, and individuals born without hands who can't imagine hand use, would allow us to deduce that what we're finding is really independent from what body part performs the action and isn't just due to some unique plasticity in these individuals. Does that make sense? Awesome. So what we're doing in the lab these days and for a short while is to try and look at how we and people born without hands perform different hand and foot actions. And for that aim, we have typically developed individuals and individuals born without hands that I'll refer to as individuals with dysplasia or IDs throughout. I'm sorry for mixing terms. It just at this point happens automatically. I'm trying to control it. But if I say IDs, this is what I mean. And we place markers on people's limbs, on their digits, in order to try and get at the parameters of their movement. Now, this is specifically a research done with my former postdoc, Yushi Liu, and my former students, James and Sri, in collaboration with Eris Freud from York University. And we were trying to look at the behavioral profile of performing foot actions. And we specifically limited this study to looking at controls with the rationale of being able to compare hand and foot actions to see if it makes sense for a shared neural plan, a shared motor plan to be driving both of them. So we looked at the trajectory of the movement, the velocity profile, and the grasp aperture. This is because when we look at hand grasping, there are several characteristics that are very common in how we perform hand action. First of all, when we reach to an object, we first have this fast ballistic stage of really moving our hand towards the object when we're accelerating. Then we reach max velocity, the maximum velocity of the movement, and then we kind of slow down towards the object. And in that stage, we can already fine-tune and control it and respond to if that object has moved or if our action has not been completely accurate. But that acceleration stage before the max velocity is considered to be a ballistic stage. It's pre-planned and can't be as affected by changing the plan. So this kind of represents the motor plan as we have it in our brains. Another thing we wanted to see was the pre-shaping of the digits. When we reach to grasp something, we will always adjust the aperture of the digits based on the object size that we grasp. And I don't know if you can see this because I have blurred background. I should have ignored that. So you can see what I mean, but we adjust the aperture of the two digits to accommodate the object that we're going to grasp. So we checked whether those principles apply the same way when we try to grasp with our foot, and none of us are foot experts, right? And yet, we seem to have a very similar acceleration phase. And you can see on the x-axis, the movement time, and on the y-axis, the velocity of the movement. In black, you can see the curve for the hand, and in red, you can see the curve for the foot, and you can see that they really nearly overlap for the acceleration stage, suggesting that that motor plan, that ballistic movement stage, is really guided by similar principles. We do have differences in the deceleration stage when we can do more motor control, and we're still looking into that, whether these are different, controlled by different regions, or whether this just represents our inexperience and inability to control our foot as well as we control our hands. When we're talking about object size, we also find that the maximum grip aperture, which is the maximum distance between the two digits, scales for the hand, as you can see, as it changes between large, medium, and small objects for the black lines, and scales to some degree, of course, not as much because we don't have that wide an aperture with our toes, but it does scale with object size with the foot as well, suggesting that these principles as well can manifest in the motor plan for guiding both hand and foot actions. Again, suggesting similar control mechanisms. And now if we want to dive into how the brain is organized, I'm going to give you a couple of slides on background of how fMRI works and what the images I'm going to show you are. So bear with me for a second. Generally, we put people in the scanner. It's a standard MRI machine, the same one you'd go to in any clinical scanning. But instead of scanning the structure of your body, we are actually providing a task or stimulation, something for you to do or see while you're in the scanner. Because what we're tracking is the hemodynamic response following that task. When we have neurons in our brain that are activated, they draw the oxygen and the glucose from the bloodstream. And in order to allow themselves to compensate for that and to draw more blood to the area, they send out signals that create vasodilation in the adjacent vasculature around them. And that increases the blood flow and blood volume to the area, which allows us to track the ratio between oxygenated and non-oxygenated hemoglobin using a signal called blood oxygenation level dependent fMRI signal, which is what we actually track in our experiments. So what we track is not a direct signal elicited from the activity of the neurons, but a secondary response that is slightly delayed in time that corresponds to the vasculature response to activation of those neurons. And what that means is that we can put people in the scanner, say control individuals like you and me, who are performing reaching and grasping actions in the scanner, or we can put individuals born without hands who are performing the exact same actions with their foot and track where in the brain is activated when we perform those actions. Now, just before I show you what that means, I'll walk you through what the images I'm going to show you are. So I'll move Daphne to the bottom here. Sorry, Daphne. What you're seeing here is a picture of a brain. You can see that it's a side view, kind of a sagittal view of cutting the brain in this direction. We can also cut the brain in the horizontal plane and have this view, and we can strip the brain from the skull, leaving just the brain. Since what we're interested in most of the time is the cortex, so the neural sheet of cell bodies that surrounds our brain, we'll try to inflate it so we get a better view of all the brain. Because if I just show you a slice, you're only seeing what you can see in this cut of the brain. So what we will do is color all these gray areas that are, cell bodies and clean them out. And then we have this very clean hemisphere of the brain, which we can then inflate. We can draw just the outline of the brain regions and puff some air into it so that we can look all the way into the depth of the soul side, the areas that are in the folds of the cortex. So those folds would be the darker gray areas, and the highlighted areas, the areas outside, would be the lighter areas. And that would allow us a view of both brain hemispheres, including all those folds. So what I'm showing you here in green, for example, this is a superior view of the cortex, where the black represents the curved areas, the sulci of the brain, and the light gray represents the gyri, the protruding parts of those folds. And the important bit is that these green areas represent the areas that care, that have a different activation pattern, depending on whether this is an action performed by us using our hands or by individuals with dysplasia, people born without hands using their foot. And CS over here would be a marker for us for the primary sensory motor cortex. So areas in and around the primary motor cortex really care about what body part is active. Using this 2x2 design, we can also look where in the brain we have a preference for the action type, whether it's reaching or grasping, which we know are different actions for hands, but are they also different actions when performed by the foot? And what you can see here, and this really should have been yellow, my apologies, this square, shows you areas that when we go away from the primary sensory motor cortex, we have a lot of areas that seem to care a lot about what action is being performed. They don't care whether it's us performing it with a hand, whether it's an individual born without hands performing it with a foot. They care whether it's reaching or grasping. And more importantly, we can even read out or decode. We can train a classifier to distinguish those actions based on the activity of the brain. So in areas that are dedicated to hand actions in us, we can read out better than chance whether an individual with dysplasia, born without hands, is performing a reaching or a grasping action, suggesting that these areas really can care about the action that is being performed and not just about what body part performs. Now, nicely enough, I kept telling you, okay, this is the primary motor cortex. These are the other external areas. And the reason I'm doing that is because what we see is essentially a gradient, where the primary motor cortex seems to represent very fine motor control and cares about what body part is acting. Whereas further away, if we go both anteriorly or posteriorly, away from primary motor cortex, we get things that are more abstract representations. They care about the action that is being performed and less about the body part. And honestly, it makes a lot of sense, right? When we think about grabbing our coffee cup in the morning, we don't think about how wide we need to have our fingers, or how much force we need, how much power we need in order to lift that cup, right? We have a way more abstract action goal in mind. And trickling down the motor hierarchy, that is translating to a fine-tuned motor command that allows us to move our fingers in the right way. So it makes a lot of sense for our brain to really represent all those levels of abstraction, anyway from, oh, I'm thirsty and I need caffeine, all the way to how much do I need those digits to be forced to apply force so that I don't drop my coffee cup in the morning. And what we're trying to say is trying to map the motor system into where we have those more abstract representations that allow us to perform the same action using different body parts. What we're doing now is trying to go beyond these preliminary findings to try and see whether this is a really broad principle of how the association action system is organized. Whether all of it really just doesn't care what body part performs, but cares about different actions or different parameters of the actions. And this is a work done with my former postdoc, Hyuchi, my students, Flo, Nama, and Caroline, and in collaboration with Jody Cullum from Western University. And we're putting more and more people into the scanner, having them look and perform at themselves, look and perform different actions with either their hands or their feet. And we try to expand on the previous findings of reaching and grasping in multiple directions. First of all, is this abstraction or this general representation for action type limited to actions that we have brain regions inherited from our primate ancestors, right? Reaching and grasping is something that so many of these animals can do as well. Would we have an action-based representation that doesn't care about the body part for things that are, uniquely human, that have evolved way later in the evolutionary tree? So now we're trying to look at different actions like tool use, which is maybe close to being uniquely human, and definitely writing, which has been around for a very brief period of time, and try to see whether that is also indifferent to the body part. Now, as a natural clue, the fact that we can all go to the beach and write with our toes in the sand should kind of give you the hint that probably that should be the case as well, right? When we write with our toes in the sand, we don't think, oh, I need to move the toes or move in this direction. It's a very fluid action. And that's exactly what we find in our studies. If we're now looking at whether we can look at tool use as opposed to grasping, so where in the brain we represent tool use, and where in the brain do we represent tool use using the hands and tool use using the foot in a similar enough way that we can use the pattern of activation for hand use to read out trials from foot use, and that's called cross-decoding. I won't bore you with the parameters. It just means that the neural patterns are similar enough between those two actions. We find that in all the areas that we have found preference for action types before. I won't again bore you with the names, but we're still looking at those association motor areas. If this is M1, primary motor cortex, we're still looking anteriorly and posteriorly from it in those association motor areas. And importantly, this is found not only in controls, as I told you, this then could stem from motor imagery, but also in individuals born without hands, because in these areas where we can decode brain activity for hand actions, we can also decode it for foot actions in individuals born without hands. And this overlap of all these pieces of the puzzle tell us that this is a general representation that doesn't care what body part is active. We're now trying to do a similar analysis for not just complete actions, but action parameters, action parts. When we think about abstraction, it makes a lot of sense for us to say, oh, of course, something like writing would be abstract, but would grasping something small versus something big be abstract? And honestly, I don't know the answer for that in a naive way that I could tell you, oh, writing with our toes in the sand makes a lot of sense to us, so this should be the case. But it does really inform of how the motor hierarchy is organized. And importantly, it has really strong implications for what we can use brain decoding in order to use that for rehabilitation, for brain machine interfaces, for prosthesis. Because if we want to use that type of abstract representation of actions for controlling prosthesis, we want to be able to derive a lot of information about it, not just, oh, this person wants to write, right? You want to know what they want to write. You want to know if they want to grasp this part or that part of the object, if they want to have a large or small grasp to reach in this direction or another direction. So this is what we're trying to do now. And we're trying to look at different grasp types, either the small part or the big part of the object, in this orientation or that orientation across the hand and foot. And again, we can find that in areas around the primary motor cortex, though also in primary motor cortex, which is interesting to us. And again, suggesting that a lot of those action parameters are encoded in a way that is abstracted away from those low-level sensory motor control programs. So what I've showed you so far is that our association motor cortex seems to create motor plans that can be used by multiple body parts in each of us. And that is what allows plasticity for the same actions in people born without hands. They're basically using the same infrastructure of the brain that we have. And the fact that we already have it allows them to use it in a compensatory manner. And then it becomes a question, to what degree does that apply to primary motor cortex, right? Because if I kept telling you, oh, association motor cortex is more abstract, we need to have those more abstract goal-type representations. In primary sensory motor cortex, what we have is a very clear topographic mapping of different body parts being controlled by adjacent parts of the cortex. And when we take out the hand area or the hand itself, what would this area do? Now, drawing insight from what animal models have told us about later in life amputation, deafferentation, a disconnection of the wires between the hand and the brain, what studies have shown is that if we have this somatotopic nice representation where different parts of cortex represent the 5 different digits of the hand, all the way from the thumb to the pinky, an amputation of the middle finger, would lead to this type of overtake by adjacent parts. So if digit 3 is no longer there, that cortical territory is taken over by the close by digits. So digit 2 and digit 4 will now have a broader neural representation at the expense of the missing digit. This could be one thing that we can expect, right? An overtake by the adjacent body parts. And in this case, most likely the arm, right? Or the shoulder, where they still have nearby body parts. An alternative to all of this is that this area doesn't just represent the hand. That much like what we saw in the association motor cortex, what it represents is something about the function of the hand, something about the fine motor control of a body part, perhaps, or dexterous action, in which case it would map onto the main body part that they use for actions. In this case, the body part they use is a hand, which is the foot, right? We've seen them. They are all foot. And I'm bringing this up to wake up someone from falling asleep, but also as a reminder that these individuals, if we have an expectation of what body part will take over their primary hand area, it should be their foot. This is the body part they use as a hand in all of their actions. Now we put people in the scanner and we had them move different body parts, and we saw where in the brain we have responses for those body parts. And the first thing is, and this is replicating studies done by additional groups before us, is that this orange blob here is the area that is responsive when we move our hands. The same area is responsive in individuals born without hands when they move their foot. Okay, we do have foot responses in their hand area. But what we didn't find is a preference for that action, right? What we needed to see is whether that area is overtaken by other body parts. And for that, we needed a more elaborate testing of what other body parts are nearby and what they are mapped onto the brain. So you don't need to have a very detailed understanding of this image, but CS here represents, and you can see my mouse tracing it, the primary sensory motor cortex. And much like that cartoon of the somatotropic mapping, we have a map of face in the bottom, then the hand, then the shoulder, the abdomen, and the foot on top. And this is a typically developed brain. So this is a group average of what our activations look like when we move different parts of our body. And then we outline the hand area in us and try to see what it would be taken over by in people born without hands. And as you can see, that white outline now is completely green and blue, completely overtaken by the shoulder and abdomen, and not by the foot, which would be purple. In other words, this area is not being overtaken by the area of the body that is used as a hand. It's not selected for foot movements. And that tells us something about the level of plasticity, right? Even their entire lives spent using their feet from an early onset deprivation doesn't allow that area to be taken over by the foot. And it also tells us something else about that level of representation in that area. What this area does can't be that high level, can't be that abstract. It has to be way more tied to the sensory motor specifics. And that is why it remains topographic and remains overtaken by the nearby body parts. So we're now trying to see whether we can use this plasticity type as a marker for the representation level that cortical regions take. We can mark in green the areas that are overtaken by close by body parts. We can mark in yellow the areas that are overtaken by the foot, which is the body part that they use as a hand. And we can plot on the brain how each body part is being overtaken in these individuals born without hands. And that allows us to draw a line between areas that are more low level and areas that are more abstract. And we hope to augment this with testing many more things about how the motor hierarchy works and what it means for plasticity in multiple levels of types of actions and motor low-level parameters, not just looking at cortex, but looking below cortex for additional parts of our brain like the cerebellum and the basal ganglia, starting to look at the temporal dynamics and trying to decode the action types and information, not just from the movement, but from the time before the movement, where we can look at the intention to act, which would be what we would want to use for brain-machine interfaces. And of course, generalizing not just across performing the actions, but also whether these same regions are involved when we observe other people using their hands or their feet. To sum this up, I've shown you that some motor execution areas and computations seem to develop for any body that can perform the same actions. This is what we call effector independence or effector invariance, and that really allows the plasticity in people born without hands. And more generally, it looks like the association motor system develops the same functions. So in people born without hands, the motor reaching areas would still be motor reaching areas just for the foot instead of the hand. And these computations can arise based on the existing connectivity patterns. I have not shown you this here, but we have evidence from that in other studies that seem to develop before we are born, before we actually rely on the sensory motor experience to fine-tune our brain structure and function. And importantly, this principle of the association cortices extending across inputs and outputs to allow us to compensate for missing inputs and outputs extends across all the models that we have been looking at, and that a lot of other groups have been as well, in blindness, in deafness, in handlessness. And it really looks like much of the brain can develop its natural functions or its typically developed functions independently from experience. I won't bore you with the details of some of my older works in blindness, but the picture is very, very consistent. I thought I'd speak more quickly, but apparently I did not. So there would be time for this, but I'm happy to talk about this later. But the picture is very, very consistent. Association visual cortices in people born blind seem to represent the same types of computations for judging shape, for judging location of things, for judging whether this is an object or a face, for judging reading, regardless of what sensory modality the information is coming from, both in us and in people born blind, allowing them to compensate for their visual deprivation. What is also interesting about the handlessness model is that it really allows us to look into the topographic early cortex, where we see a very different organization principle, right? What we have there is an area that is way more tailored, or way more fine-tuned for the sensory motor elements themselves. And that brings us to think about how those hierarchical structures and cognitive capacities exist and develop in the brain. And in truth, what we expect to find is that there are multiple levels of the motor code represented at same and different times as the brain is activated in sort of a mixed neural code in areas that would care about both what body part is acting and what action is being performed and what action parameters are being performed, simply because we have so many levels of things we want to control when we perform an action. And the question is open to me whether this can be used to drive better prosthetics. Theoretically, at least, we can use some of that understanding of the more abstract level of representation and motor plan to develop better control of brain-machine interfaces and prostheses that are currently more tailored to augmenting and deriving those low-level muscle pattern readouts that don't differentiate in advance whether this is a grasping action or what is the size of the object you want to grasp, and focus on fine-tuned motor control, which is harder and less natural for us to do, because when we try to act in the world, we really don't think about this and we don't represent it as much as those high-level goals. And interestingly enough, this could also have some relevance for rehabilitation from stroke if we think about training not just the hindered body part, but the hindered action type that is being affected, and training it by unaffected body parts that don't have that motor limitation. I'm also happy to talk to you about blindness. We have a partly unrelated research line looking at individual differences in brain plasticity, what the role of environmental factors and experience are in driving these, and what it can mean for sight restoration. And more importantly, I'd like to thank all the people who have contributed to this work. Some of the earlier work, the somatotopic ones, were done with Gilles Manuscore and Alfonso Caramazza back when I was at Harvard in my postdoc. The current works were done with Erez and Jody, and multiple people in my lab, especially Yuchi and Flo and Nama, who have been instrumental for these works. And thank you for your attention.